metabolism, the definition of it is the sum of all the chemical reactions that occur in the cell or in the human body. So the collection of all those chemical reactions, right? The chemical reactions to make your muscles move, right? The chemical reactions to make your guts move and digest food, to pump blood around your body, right? To think. Those sum total of chemical reactions are called metabolism. Now, in chemistry, this is kind of an empirical equation for a chemical reaction. In grade 10, we call this a single replacement reaction or double replacement reaction. You see that? A plus B making C and D. It's very general. In this, we'd have the stuff going in. What do we call the stuff going in grade 10 into a chemical reaction? Yeah, the reactants, right? In biology, we call it substrates. Usually we use the word substrates in biology. And then the arrow is yields or leads to or makes the products, and that's the same. Okay. Now in biology, we have two types of chemical reactions. We have a chemical reaction that puts things together, right? So when you eat food and you digest it, you'll break it down so that you have amino acids, so that when we make proteins using mRNA, the ribosomes can add those amino acids together. So adding them together is called an anabolic reaction. Okay? Adding anabolic. Now, to get those amino acids to build them together, we have to eat food and we break them apart. And that's called a catabolic reaction. So we have two types of metabolic reactions. One's called anabolic and one's called catabolic. Now, if you take your finger and you shove it up your nose, you'll find that it's not super warm. Okay, you can try it if you like. You can shove it in your mouth too or your ear, and you'll find that it's not really super duper hot. And to be honest, that temperature, it's around 37 degrees. Some people are slightly lower their basal body temperature, and some people have a slightly elevated basal body temperature. So that means that when they're resting, they're not exercising, they're not super in a cold environment or a super hot environment. Their resting temperature is around 37. And you'll find that that's not hot, right? You shove it in your mouth, you don't burn your finger, right? When you pick your nose, it doesn't get really, really cold or hot. It's just 37 degrees, right? Relative to ambient temperature. And if you're in your house and you like it warm, you turn your thermostat up to 23, right? And you walk around in your underwear. Or you turn it down to 17 and you bundle up with socks and sweaters. But it's around 20 degrees outside. So you're a little bit warmer than the outside, but you're not flaming hot. And to be honest, that is not the best temperature for chemical reactions. If you're in chemistry class and Mr. Morrison says, oh, do this, and you're like, nothing's happening, he'll say, warm it up, heat it up, see what happens, right? And that is because most chemical reactions have something called the energy of activation, right? It's the amount of energy that you have to kick it to get it going. Now, you might say, do all chemical reactions need a kick? Most of them do. It's kind of like a rock at the top of a hill. And you're like, oh, if that rock ever started rolling, it would just roll down that hill, no problem. But it's not going to roll by itself. You still have to go and nudge it, right? And that nudge that you give it is the amount of energy you put into it to get it going. And after it's going really well. Now, other rocks, right, you might have to push it right up the tall side of the hill just to get it back down the other side. And that one would be a huger or larger amount of energy required, right? And we call that the activation energy. Okay? Now, if you look, this is the list of things that we need usually for a chemical reaction to happen in the body. Temperature, and we already told you it's about 37 degrees. pH. In the human body, the pH is around 7. Blood's around 7.4, right? And you might be surprised, but your stomach is around pH 1. It's super acidic. And your other parts of your gut's around 8, right? It's really basic, fairly basic, I mean. And then you have to have stuff to join together, obviously, or break apart. And there's the activation energy, the energy to start. And then enzymes, because like I said, the temperature in your body is not warm enough for most chemical reactions. If we just got a bottle of water and we mixed it in some sugar and all those things that are inside your amino acids, lipids, and we put it into a glass of water and just left it at room temperature, nothing would happen, right? We'd have to heat it up or something like that. Now, in chemistry, we can speed up a chemical reaction, right? What's the chemical that we would use, the substance we use to speed up a chemical reaction? Anyone remember that from chemistry 10, or if you took chemistry 11, you might remember it? It's a catalyst. So a catalyst is a substance that speeds up a chemical reaction without being used itself. Well, we have organic catalysts. So they're made of carbon, hydrogen, oxygen, right? And those are called enzymes, usually protein. These guys will speed up a chemical reaction at pH 7, temperature 37, without being used up itself in the chemical reaction. Now, enzymes, organic, do they last forever then? No, just like the milk you left on the counter, it will go bad. Okay, so if you need this chemical reaction to happen, the enzyme will come in and do its job. But if you need it to keep happening, you have to make more enzyme. And that's how your body controls how much of a reaction needs to be done, how much product needs to be produced. Because your body will produce product, and it will say, hey, we don't need this much. And it'll be, oh, that's fine, just wait a little bit, because the enzyme's going to degrade, and the reaction's going to fall off. And then your body's like, oh, we need more of that product now. And your body's like, oh, has all the enzyme disappeared? I'll make some more then. And then it'll produce some enzyme to rise the rates again. Okay? So let's take a look at that activation energy. Activation energy is the amount of energy required for the chemical reaction to start, to initiate. The symbol for it is this weird E sub A, capital E subscript A, E sub A. Okay? Some people call it the activation energy. Others call it the energy of activation. Doesn't really matter. I guess it's like French and English, right, when they put the adjective in the wrong place. So there you go. This is the typical graph of one. So this is the progress of the chemical reaction, so how it's proceeding. And this is the amount of free energy in it. So for this chemical reaction, blue, I have to put in that much, and then look how much I get out of it. So I have to get it going. A good example is this. If you go camping and you want to start a fire. You're like, oh, we need to start a fire. Go get firewood. And you take your firewood, you put it in the middle, and you go, it's not warm. Like, what's going on here? Well, what do you need to do? You need to put some energy into that chemical reaction first. You need to light a match, right? You got to get some paper going. You got to raise the temperature a little bit. And then once it's burning, you don't have to do anything anymore. It's rolling all by itself. Similar to trying to roll a rock down a hill. Sometimes you have to roll it up the hill a little bit, and then it'll roll down the other side, right? Now, we can lower the temperature for energy of activation by putting in an enzyme. So when you put the enzyme in, look how shallow the hill is now. So we still have to give it a push, but it's not a huge push. It's a smaller push. And in this example, it's about half the amount of energy to get it going. But once it's going, it still goes. Yeah? So this graph could represent two things. It could represent two different chemical reactions where one requires a little bit of activation energy, one requires a lot more activation energy. Or this can represent an enzyme present, and this is what it is. So this is the same chemical reaction, we're just putting in a catalyst. And this catalyst is letting it happen at a lower temperature. So what is an enzyme? An enzyme is a protein. We call it a biological catalyst, and it speeds up a chemical reaction without being used in the chemical reaction itself. It will change over time because it's organic, and it will die, right? It will just run out of, it'll just denature and run out of usefulness. And then your body will absorb it, probably digest it, and then put it, and maybe make it a new enzyme if it needs more later. You can tell that it's an enzyme because most enzyme, they end with this funny terminator A's. So who here likes to have some ice cream, but their belly isn't really fond of it sometimes, right? Yeah. Sometimes I don't I'm not a milk fan. I like ice cream once in a while, but sometimes you'll get like a hot day and you just drink a whole bunch of chocolate milk, and then later on you're like, holy smokes, you shouldn't have chocolate milk, right? I got a bit of the runs. Well, that's called irritable bowel syndrome sometimes. Other people call it lactose intolerance, because you're missing an enzyme in your body to help you digest lactose. That's a milk sugar. And the milk sugar that travels through your belly just irritates your bowel as it goes through, and it just kind of makes your bowels want to get rid of it faster. The enzyme for lactose is lactase, right? So the A's tells you it's an enzyme, and the lact tells you it's for lactose. Another one is, if we're going to talk about digestion later, you'll eat foods, and one of the things that's not digested in your stomach chemically is fats. They get digested later at the duodenum by help of the pancreas and a little bit by the liver. And fats have a nickname of lipids. So anyone know what the enzyme name for digesting lipids might be? Yeah, lipase or lipase. Yeah, there you go. So there you go. Lipase for digesting fats, lactase for digesting lactose. And then there's a whole bunch of other ones. We'll look at them, like peptidase, proteases. There's lots of them. This is what an enzyme kind of looks like. You got a blue enzyme there, and you got an orange enzyme here. This one is breaking stuff apart. So what type of metabolic reaction is that called? Breaking apart? Huh? I can't hear you, sorry. Yeah, it's a decomposition reaction for sure. But when we talk about metabolism, we have two special names for them. Catabolic. It's a catabolic reaction, and the chemical reaction is actually called a decomposition reaction or a hydrolysis reaction. Okay? But it's the catabolic part of metabolism. This one's putting stuff together, adding it together. So that's the anabolic arm of metabolism, putting stuff together. And anyone know the chemical name or the chemical process in biology that puts things together? It's got a really funny name. In Science 10, it'd be synthesis reaction, yeah, when you put two things together. In biology, it's called dehydration synthesis, yeah, because water is actually one of the products when you put them together. Water gets cut out, okay? So this is catabolic, anabolic side of metabolism. This one requires a chemical reaction called hydrolysis or decomposition, and this requires a chemical reaction called synthesis, or dehydration synthesis, okay? So here's your enzyme. Enzymes work by taking substrates and holding them in a perfect alignment so that they can be broken or joined together. By holding them in perfect orientation, they require less energy, okay? So if you had these two guys wanting to join together and they're just randomly running around, they can fly by each other and miss each other, or they can just kind of glance and strike off of each other at an angle. But if they were to come straight at each other and bind, they need a certain amount of energy to do it. Now, if you have an enzyme doing it, an enzyme does it by just taking them and then aligning them perfectly, and then bang, it's so much easier. So the amount of energy required there is way less. The analogy we like to use is, you know, everyone's heard this story. We all have a match in this universe. Hopefully it's not any further than on the Earth, because like if you're supposed to find a match that's an alien, good luck. But let's pretend we all have a match here, and you're supposed to you know, hopefully someday find that person. You can see the match will be very difficult if they're outside of this solar system, outside of this planet. But let's pretend it's on this planet. Still really difficult, isn't it? This dude or dudette could be walking around on, you know, the other side of the planet, not knowing that their perfect match is here at McGee studying A&P 12, right? We could make this story a little bit easier. We could say that this person is in the school, right? Now, in order for you to meet your perfect match, it would have to be, right? You might walk by this person every day this quarter as you go from one class to the, you know, to go home, or coming into the school and they're going out. It could happen. You would never know. You need perfect alignment. Now, it also requires another thing, because if you're just walking by and you just kind of like nudge each other's shoulder, you're just like, oh, what was that? And you just keep walking. That doesn't work either, right? You need to come face to face. You know, in the movies, it requires direct eye-to-eye contact, right? And then there's like this energy, and then all of a sudden you live happily ever after. Or you could have a matchmaker. And the matchmaker, right, could be your best friend or someone that you know, and they're like, I know someone. And they grab that person's hand, and they grab your hand, and then they put them together. Perfect alignment, less energy. That's what an enzyme is a matchmaker. Okay? And as a result, it requires less energy. Now, how do they work? There's two accepted models. One's called the lock and key model and the induced fit model. They kind of work hand in hand. But what we need to do is we need for the enzyme, right? We need the enzyme to somehow find the substrate. Now, on an enzyme, you may remember this. We started off with DNA. We said this is a gene. We're going to transcribe that using RNA polymerase. Oh, look, RNA polymerase pooped out this mRNA. And the mRNA's got some bad sections, so let's just cut out those introns. And now this exons that are joined together can leave the nucleus and go into the cytoplasm. And in the cytoplasm, we've been using this thing as the ribosome, right? And the ribosome will read it and add amino acids together. And you get a polypeptide chain. So that's a primary structure. Now, the primary structure will do this, which is secondary folding. We call it alpha helixing, where it spirals like a piggy's tail. Or it can do this, where it goes up, down, and it turns into a sheet. And we call this a beta-pleated sheet. So that's secondary folding. And then what can happen is, let's just pretend that this one does that and this one does the loop. Right? So you got, but it doesn't have to be. It could be actually a bit of looping here and a bit of sheeting there. So it can be quite long and complicated. Now, this thing can do this thing where it falls upon itself, and we call that tertiary folding. And then when you have 2 or 3 or 4 subunits come together, that's called quaternary structure. That's the final structure. And in that final structure, there's going to be a spot where the substrate, I was using this dice, this substrate fits in perfectly. And that site is called the active site. Now, if there is a mutation in here, point or chromosomal mutation, and that led to a mutated RNA, that would lead to a mutated polypeptide chain. So when it comes together, guess what? That might be slightly different. And this substrate that comes along will go, oh, I'm supposed to fit there, but it's not quite right. So I'll just keep trucking along. So does this substrate ever find the active site? No. And that's what mutations do. They stop. Often they'll just be enough to make it so that the activation site, the active site, isn't working anymore. What we want is the substrate to bind to the active site, and what we get is, it's called enzyme substrate complex. Okay, and that holds it in perfect form. And what can happen now, and this is a catabolic reaction. It can break it apart. Okay, in there, and then once it's broken apart, it can release. Now, this only happens though. So the lock and key model, if it is perfect in the active site. It has to be a perfect fit. It's like a lock fitting into a key. If it doesn't fit, you don't get to turn it. It's got to fit perfectly. That's why it's called the lock and key model. And this is a picture. See the E? That stands for enzyme. And then you got 2 substrates coming in, and they fit lock and key. Check it out. Now, is this a synthesis reaction or dehydration, or sorry, a hydrolysis reaction? It's a synthesis reaction. Is this the catabolic side or the anabolic side of metabolism? It's anabolic, yeah. And you can see, matchmaker, hold their hands, introduce them, and union. They're joined. Enzyme releases is not changed at all by the chemical reaction. It might change because it's organic, so it's going to decay over time. But now it can go do the same thing. You can go be a matchmaker to another stripy and a black rectangle. So if you can find those 2 substrates, the matchmaker can keep doing that over and over again until the matchmaker unfortunately dies, passes away. Now, Induced Fit Model is a little bit along the same lines, but it says that the substrate enzyme complex allows for the enzyme to actually hold it. So let's pretend that you do have a match, right? And your matchmaker friend comes, I know, I've seen your match. I'm going to go get them. And it happens to be grade 8er, right? Kind of creepy, but it's a grade 8er. And the grade 8er doesn't know, right? It doesn't want or doesn't understand. But when you get older and your ages kind of come closer together, right? Right now it's like, 13 and 17. But when you're 30 and 28, it seems closer and closer. So to get that grade 8er to introduce you, you're going to have to grab them and hold them. And that's what the induced model is, right? There's a little bit of we call it a conformational change, which locks it in place so it can't run away. Now, there could be some introductions, right? Probably the grade 8er will run away and you'll be like, whatever. We'll see you in about 20-something years or something. But the induced fit model is that the enzyme substrate complex is lock and key, but also a little bit more induced fit so that it holds on. It allows for better alignment, right? So the activation energy can lower. And once that catabolic or anabolic reactions happens, it'll change shapes, release it, and then do it again. It's like Pac-Man. Okay. Any questions so far? So those are the two accepted models of how enzymes work: lock and key and induced fit model. There's some other things though, right? You may have heard of vitamins, right, and minerals. And you might say, why do we need those? Well, vitamins and minerals help enzymes work. So they're called coenzymes or cofactors. If it's organic, it's a coenzyme, just like enzymes are organic. Coenzymes are organic, like vitamins, right? Vitamin A, vitamin C, vitamin D. Cofactors are inorganic, like zinc, right, and copper. You see those people, they like to wear their copper bracelets. Right? They think they absorb copper and it makes them healthier. Zinc, if you ever get a cold or flu, sometimes people take zinc lozenges, and that's supposed to help you recover from colds and flus faster. Iron, you need iron in your diet, right? If you're a vegetarian and you rather pet your animals than eat them, sometimes what will happen is you'll become anemic, right? And they'll tell you that you need to have more iron. So if you're not eating meat, maybe you just have to drink the blood of an animal. That, or you eat those really dark green vegetables, which are heavy in iron as well, or you take an iron supplement. But that's a cofactor. So how it works is enzyme E, see it? Substrates, gray. But as you can see, gray does not fit into the enzyme E unless the coenzyme fits in there as well. Right? And you've heard of disorders where there's not, when you're lacking a mineral or a vitamin, such as scurvy and rickets. Right? Those guys that would come over on the boats and not have any vitamin C, and then they get scurvy and their bloody gums and stuff like that. When you go off to university, your first year, there'll probably be a story about this. I remember the story where a boy or girl moves away for the very first time to live on campus, and all they eat is potato chips and Kraft macaroni and cheese. And then they never leave their dorm room. They just play video games and stuff like that. And then you'll hear stories of some kid developing scurvy. Every year we heard the story about some kid in dorms getting scurvy because they weren't eating the right things, right? You're missing a vitamin or a mineral. You can develop these disorders. You might get all the nutrients you need, but you're missing those vitamins and minerals. All right. I want to give you a break, but I did want to show you one last slide before you take a break. And it's metabolic pathways. Okay, we talked about what metabolism is. It's all the chemical reactions in your body. Well, metabolic pathways, lots of us, like in chemistry, we go, just add this with this and look what you get. It's just like a single step. In biology, they're often multi-stepped. So if you need product D, it's going to go through B and C using enzyme 1 and 2 and 3 before you get to D. Okay, and there's some good and bad to it, right? the fact is that some of the good parts is that you can store some of the stuff as B and C before you get to product E. Okay, so you know, if you eat a whole bunch of nutrients and you don't need it right away, we can store it, and then it can lead to that later if we turn on 1, 2, or 3. Another thing is if you're overproducing D, we can turn some of these off so you don't overproduce it. Right? So there's a whole bunch of control factors in there. But you can see if you have I guess a defective gene or something and it's not quite working, then you can kind of screw up the rest of the pathway though as well. But most metabolic pathways are multi-step, requiring multiple enzymes. Okay? So just to summarize, A to B leads to C and finally D. Each change requires its own enzyme.